Hello, everyone, and welcome to Plant Rhizospheres by Richard, Kiana, and Jamie. So we're going to hop right in. This is the introduction. So the first picture here is a little bit on the influence plants have on the environment, with the rhizosphere being an important part that deserves more attention. With increasing pressure on the agricultural industry to produce more food for the exponentially growing population, scientists have returned to the roots of food production, the soil. The soil, despite its dull brown appearance, is the lifeblood of survival for a vast majority of organisms. Without that soil, within that soil, the rhizosphere is a bustling region of activity, influencing landscapes and global processes. Understanding these processes are critical to ensuring that plants grow up and are able to thrive, and in turn, enable us to feed our world. So what exactly is the plant rhizosphere? The plant rhizosphere is a section of soil that is present in the immediate environmental vicinity of plant roots. The specific section of soil has a unique composition of microorganisms that are not present elsewhere in the plant or the soil. The microcosm is highly specialized and flourishes with a complex symbiotic relationship where plants and microorganisms interact and influence each other's growth. The microbiome that the root creates is fluidly defined by plant type, climatic condition, environmental stress, and a variety of other factors. However, the microbiome can also affect those same factors. To give a quick overview, our lecture is going to be split into three parts: rhizosphere development, biological significance, and the mechanisms.
Hello, I'm Kiana, and welcome to the first lecture where I will be speaking upon plant rhizosphere developmental processes. First, let's begin with root exudes. Root exudes are important to a plant because they effectively recruit a diverse set of microorganisms into the rhizosphere, as root exudes are often a source of carbon and energy that most microbes rely upon. Root exudes are often found to be various sugars, amino acids, growth factors, vitamins, or other chemoattractants. So how do root exudes play a role in the formation of the rhizosphere? Well, root exudes coincide with the recruitment of microorganisms, as there's a complex network of communication and interaction between a plant's roots, root exudes, and recruited microorganisms. To begin, a plant begins its recruitment as a seed or a cutting placed into the soil. As roots emerge, they begin to interact with an already existing microbiota in the soil, thus the formation of the rhizosphere. As roots produce exudes, diverse microbes are recruited into the rhizosphere, thus generating the complex network we know today. We now understand that the root exudes recruit microorganisms into the rhizosphere, which explains the large amounts of diversity within this area. However, there is still some ambiguity around the kind of microorganisms that can be recruited. In this experiment, led by Thomas Cost, it was discovered that a specific bacterium colonized on lupine and maize plants. This bacterium, B. phytofermins, had the innate ability to break down oxalate and use it as a carbon source. I say that this is innate because this trait was only found in this species in particular and not any other species found in this genus. It was found that B. phytofermins colonizing the plants because they were releasing higher concentrations of oxalate in response to a low nutrient, high heavy metal soil concentration. So we see that the relationship between this bacterium and the plants was found to be beneficial to lupine and maize, as lowering the levels of oxalate had a strong correlation with the plant protective feature. To confirm this, Thomas Cost and his crew created a mutant form of the bacterium that was generated, in which the gene that produced oxalate degradation was made null. The experiment was repeated, and the results showed that there was a less colonization of this mutant to lupine and maize plants, and that these plants showed reduced protective potential. So what we can take away from this little experiment is that it shows that there is a heavy correlation between root exudes and the type of microorganism recruited to the plant. As in, a plant can affect the type of microbe recruited by altering what it secretes. This sort of specialized recruitment was also shown between various stages of Arabidopsis development, with early stages of Arabidopsis plants exhibiting higher levels of sugars secreted within the roots early on in its development, while in later stages of the development, there was less sugar secreted. Consequently, early stages of development had low levels of amino acid and phenol secretions, while later in its development, there were higher amino acid and phenol secretions. This now leads us into our question of the day.
Why would root exudes change in composition as the plant develops over time? And what does this imply about plant rhizosphere interactions?
Hi, guys, and welcome back to Lecture 2, the biological significance of the rhizosphere. So as Kiana previously stated, exudes can be very, very complex or very basic organic macromolecules. They are specifically important during the responses to nutrient deficiency. One method by which plants respond to environmental stresses regarding lack of nutrients is to alter their root morphology and change the chemical environment of the rhizosphere. Such an example is to change the acidity to allow greater intake of nitrogen. Many plants use this to create a niche environment for microbes, of which we will look closely at too. The first one is legume rhizobia. So legumes, such as peas or beans, have been historically known to enhance soil fertility when grown in rotation with other crops. This is due to microorganisms that grow within the plant, as they are able to convert atmospheric unusable nitrogen into ammonia, a usable form of nitrogen. This is catalyzed by a rhizobia bacterium that forms nodules formed in plant roots. When legumes are grown in nitrogen-poor conditions, they release a flavonoid molecule. These flavonoids induce NOD genes, or NOD genes, which can go through a signaling cascade to allow bacteria to grow within the root. Plant and rhizobia interactions are extremely specific, and only certain species of rhizobia are able to grow within certain plants. Specificity that comes as a surprise when a single gram of soil contains more than 10,000 different species. The next thing we'll look at is mycorrhizal fungi. So mycorrhizal fungi are the fungal portion of the symbiotic relationship between fungi and plant roots. Unlike rhizobia, however, mycorrhizal fungi are not specific and appear ubiquitously across a variety of plants. The fungi assist the plant in assessing and accessing nutrients such as water, phosphorus, and other micronutrients. In return, the plant provides fixed carbon for the fungi to use. There are two forms of mycorrhizae, ecto and endomycorrhizae, dictated by how they interact with plant roots. Ectomycorrhizae are usually found within woody plants, such as trees or shrubs, where they form a layer of hyphae around the root itself, growing in the intracellular spaces between the cells. Endomycorrhizae, however, grow within the cell itself, forming a arbescule, although it remains surrounded by a plasma membrane. Regardless of the type, both types are able to increase the surface area between fungi and plant, and then fungi and the soil, to facilitate the transfer of nutrients to the plant. Mycorrhizae have also been shown to detect the presence of carbon dioxide in the soil, which activates hyphal growth, and similarly, plants detect mycorrhizal factors, which are also named MYC factors, which activate root branching and growth. For the next question of the day.
So the same species of plant grown 5 feet apart was found to react similarly when nitrogenous fertilizer was applied to only one of those plants. How is that possible?
Welcome to the third lecture, and we are going to cover the mechanisms. So the rhizosphere area is the narrow region of the soil influenced by associated soil microorganisms and root secretions, which is located immediately adjacent to roots. The rhizosphere has been described to include 3 different zones: the endorhizosphere, rhizoplane, and the ectorhizosphere. The soil outside the rhizosphere after passing the ectorhizosphere is the bulk soil, where it is not penetrated by plant roots. Symbiotic nitrogen-fixing bacteria such as legume rhizobia and mycorrhizal fungi, as Richard mentioned before, they detect compounds like flavonoids secreted by the roots of leguminous plants and then produce node vector that signal to the plant that they are present and will lead to the formation of root nodules. These bacteria are sustained by nutrients from the plant. Here is nitrogen fixation. Nitrogen-fixing bacteria living in legume root nodules convert atmospheric nitrogen into ammonia, which is available form for plant to uptake. Then through the ammonification and nitrification, ammonia converts to nitrites, then nitrates, so that it could be reuptake through assimilation of plants, or it can be converted back to atmospheric nitrogen, denitrifying bacteria, so that it can hop on the nitrogen cycle again. Plant rhizosphere interaction and its developmental processes are used in various ways to control the growth of the crop and to prevent plant disease. We can increase or decrease the microorganisms populations by controlling plant exudates or introducing new organisms. By using actinomycetes, which is abundant in rhizosphere, it can serve as antibiotics that could impact on plant and microbe interactions like nodulation, and it can biocontrol against plant pathogens.
So question of the day for the last lecture. I want you to review all 3 lectures for the first exam and think about why plant rhizosphere interaction is important in plant and why it is important for us. Okay, that was all 3 lectures summed up in one. Thank you very much. We hope you, I messed up. Let's try that again. Let's see, what else?
Yeah, we can change the color.
Thank you. Thank you.
Thank you.
Thank you.
Okay.
And then our resources.